summer, Jack Ahrens worked at the National Institute of Health under the mentorship of Dr. Robert Crouch. And while there, he worked on 4 different projects relating to the RNase H protein families. And today he'll be presenting one of those projects where he successfully mutated and cloned a RNase H2. Thank you, and please welcome Jack Ahrens.
Hello, I'm Jack Ahrens. Daniel did most of my title slide for me, but I worked at NIH over the summer, and this project I'm about to focus on, in this, I mutated RNase H2, which is a type of this RNase H protein family. And I mutated the yeast version of RNase H2 to create what could potentially be a hybrid deficient mutation. So before we go into my actual project, just for some background, we're going to first need to understand the concept of RNA, DNA, and YOMA. So in this process, you have these double strands created of half RNA, half DNA. And as you'd expect, when you think of a double strand would be just DNA, and then RNA, especially in the mRNA form, is just these single strands. But this is actually fairly common in the cell. And the 2 most common causes of this are going to be replication and, in the case for primers. So during replication, as some of you may know, you have the DNA strand is split up into half, and then each strand gets deoxyribonucleotides added to it in procession. But every once in a while, which is a rare occurrence, but not that rare considering how many DNA molecules are added, you have these RNTPs, these single RNA molecules added into this strand. And then another occurrence where this happens is for primers. So for replication, the primer that sets this process off is actually an RNA primer. So this single strand of RNA will yield to the origin replication of the DNA. And what happens is that you get these things called R loops. And R loops are an occurrence in the DNA when you get too much RNA incorporated into it, and you end up with this triple strand. So the DNA double strand breaks off, and one becomes its own single strand of DNA, and the other becomes this RNA-DNA hybrid in the middle of the DNA. And this is unstable and can lead to mutations. So you want to get rid of these. And if these are left unchecked, there's a condition called AGS, Aicardi Goutieres syndrome, which is genetically inherited, and it's a neurodegenerative disorder that leads to a shorter life expectancy and severe motor impairments. So RNase H is a family of proteins that have the job of getting rid of these RNA-DNA hybrids. And they're found in almost all forms of life, and there are 3 forms, H1, H2, and H3. Now, we really only study H1 and H2 for the most part, and this is because these 2 proteins are found in humans. The H3 protein is found in some forms of bacteria, but the applications for that are fairly limited for medicine. And RNase H2 has 2 separate functions, and this is important because for my project. And these 2, you should remember, these are 2 independent functions. They might sound similar, but they're carried out by the same protein, but different parts of the protein, different processes. So the first one is RER, ribonucleotide excision repair. And in this is what happens when you get these single ribonucleotides incorporated into the DNA. So RNase H2 will go in with a few other proteins, take out this single RNA molecule, and then put in a new DNA molecule. And then its other function is hybrid digestion. And this is when you get these longer strands of RNA and DNA. And this is a completely different process, and it will go in and just break up this long strand. And another important thing you need to remember is RNase H2 was made up of 3 different subunits, the A, B, and C subunits. So these are 3 proteins that are actually created separately, but then they meld together to form this RNase H2 protein. So the purpose of my project was to mutate and clone the Saccharomyces cerevisiae, which is yeast, version of RNase H2 to create what could possibly be a hybrid, a version of this RNase H2 protein that would retain this RER functionality, but not be able to do hybrid digestion. Now, the specific mutation that I did had been designed but not created by a previous fellow at the lab. So my job was to go in and actually create this mutation and express it, create this mutated protein. And this diagram right here is just another illustration of what I was talking about on the last slide. You have a RER right here, which is these smaller single ribonucleotides in this larger DNA molecule. And then for RNA-DNA hybrid processing, these are the longer RNA-DNA double strands that occur. This is a common occurrence when primers get stuck to DNA. So to carry out this mutation, site-directed mutagenesis method. And the first step in this is to order oligos. Oligos are just short strands of DNA with a specific sequence. And this sequence for this mutation is going to match the DNA almost perfectly, but in the center, it's going to have a few deoxyribonucleotides modified to be the mutation that you want to get in the result. And these act as primers. So they stick on your template strand of DNA because the sequences match up with where you want it to go, and it just has an affinity for it because the sequences match. But the stuff in the middle is not going to be the same, so I'll go into more detail later on how that works. So then you use these oligos in a PCR reaction, polymerase chain reaction. This is a common process used in pretty much any lab that studies DNA. It amplifies DNA exponentially. And then the template strands will then be digested by DPN1 nuclease because they remain methylated. So the template strands start out methylated. It's just naturally occurring in E. coli plasmids. And then, but the new DNA that you create is going to be unmethylated because it's just been created. So this DPN1 will only target the methylated DNA. So here's just an illustration of how this PCR works and how quick change PCR specifically works. So the PCR components you put in, you have this DNA sample, and these are plasmids I had that had this yeast RNase H2 protein on it, coded in it. You put the primers in, but the primers are a little different. So with typical PCR, the primers match exactly to the template strand, to a piece of the template strand. So you get the template strand copy exactly. They're going to be a little different, and the difference is the mutation you want. And then you put nucleotides in because these get incorporated into the brand new DNA strands you get, because you need to add these new DNTPs to actually amplify this DNA. The Taq polymerase is the protein that goes about creating new DNA strands and carrying out this replication process. And then you have mixed buffer and PCR2, and these are just to help the procedure go. So what happens in a PCR machine is it's really just a temperature cycle. So you have this strand of DNA right here, and once you bring the temperature up, the strands break apart. And once this happens, the polymerase, the Taq polymerase will go in and add these new DNTPs to each side, and all of a sudden you have twice as much DNA as you had before. And if you just let the cycle, I did like 30, 40 times for different procedures, you get exponentially more DNA than you started with, and then you can actually use this for sequencing, you can use it to study. It's much easier to work with. And this is the quick change process right here, just illustrated. And if you start, this colors are a little hard to see right here, but this is going to be green and orange. And the two primers are these arrows, with the red X's representing the mutation. So these anneal to your template strand of DNA, template plasmid, and it's amplified through PCR, except the template strand stays methylated, while the new strands stay unmethylated with the mutation, because the Taq polymerase goes over the primers that you used, and the primers have the mutation on. And then once you digest the methylated template strand of the DNA, so it's no longer there, and in theory, you're only left with the DNA you want, the mutated DNA. So I use this quick change kit to delete one codon in the C subunit of RNase H2, and I changed another one to create this mutation. And afterwards, I transformed this DNA into E. coli cells. Now you might ask, why E. coli cells when this is a yeast protein? Well, in the lab, you use E. coli for many different things, but one of the common uses is to store proteins like this. And with a template DNA plasmid, which is just a circular loop of DNA, you can put it in E. coli cells, and they can produce proteins that you want, no matter what organism it's from. And you can also store these, and as the E. coli, they're very easy to work with, and as they multiply, they take the protein with it, they replicate this protein. So I grew out these E. coli colonies I got from the quick change after I transformed them into the E. coli cells. And I ended up with 4 colonies, which I labeled A, B, C, and D. And I sent each colony in for sequencing of the target region, which should have the mutation. And it turned out from this that only the D colony actually had the mutation I wanted. But there was a problem here too, because there was a mix. So there was a mix between around 60% of the mutated DNA and around 40% of the unmutated template strands. So obviously what happened is that the DPN1 I added later did not get rid of all of this template, which is a problem, and it shouldn't have happened, but it makes something went wrong in the mixing process. So there are either two situations that could have caused this. So So either two plasmids got incorporated into the same cell, which is very rare, but could possibly happen. So the template and the mutated DNA got into the same cell. Or what happened is that when E. coli colonies grow, they usually start from one cell. So what must have happened if the double plasmid theory didn't work is that two different E. coli cells actually started the same colony together and meshed this DNA, which is rare because these plates, there are only 4 colonies growing on pretty separated parts of the plate. So I went about 3 ways to try to get a pure colony of E. coli that would have my mutation. So the first one was to restreak the D colony cells, and this is just taking a toothpick to the cell, streaking it out on the plate again. It separates the E. coli, lets them grow, and it just further isolates each cell from each other. Another one was to retransform the mutated plasmids. So I had more plasmids that I hadn't put into E. coli from my previous quick change. So I went about putting these into a new strain of E. coli, growing these out and seeing what I got. And then another one was to repeat the quick change process all over again. And I did this in case you had this plasmids. But this wasn't quite necessary, but it was more of a learning experience. So to continue the methods, I sent each 4 colonies from each method in for sequencing, and I ended up with actually only 4 pure colonies. So 3 of these were from the retransformed cells. 1 of these was from the retransformed cells, and 3 was from the restreak cells. Now, the good thing about this retransformed cell is that I used the BL21 E. coli strain. This is a synthetic strain of E. coli, but it's designed specifically for targeted protein expression. So you put a plasmid in, it won't express this protein unless you want it to. And I wanted to be able to target this RNase H2 to be expressed. So I ended up using the retransformed E. coli to express the protein. And I did this by adding IPTG solution. It just induces this protein expression in the cell for the BL21. It's not too important what it is, but so I induced these cells to transcribe it, and then I extracted all the proteins from the cells and ran a protein